Today we'll be talking about the structure of chromosome. First, to understand the structure of chromosome, we need to understand that chromosome is made up of two things, that is chromatins. Chromatins, if this is a chromatin and again another is a chromatin, and this chromatin plus chromatin they gives to a chromosome. They give rise to a chromosome. Chromatin plus chromatin will give you a chromosome. Now, how are they made up of? To understand the chromosome, we need to understand. To understand the chromosome, we need to understand what is this chromatin. Actually, this chromatin is made up of two things. We will understand that these chromatins are made up of two things. Number one is histone. Number one is histone. Another is made up of DNA. DNA. This DNA containing in the chromatin is of 40%. Is of 40%, and the histone is 60%. Histone is 60%. Okay, to understand these chromatins, we need to understand how does this histone look like. Actually, histone is a spherical things. It is just a spherical protein. So in exam they may ask you, name the protein that is present in a chromatin. So answer will be histone. And now suppose if there is an 8 histone. Now this is under histone, and this is also another histone, and this is under histone, and this is under histone. Sorry, maybe this is under histone. So how many histone are? 1 2 3 4 5 6 7, and let me add here 8, one more. So 8 histone. In this 8 histone, each chromatin there will be so in this 8 histone there will be a DNA. Now let me take the color DNA. The DNA will look like something. The DNA will be wrapped in here. The DNA will be wrapped in here in the histone. It will be wrapped in the histone, and histone, so they are wrapping in histone now here. And the length of this DNA is about 2 meter. 2 meter. The length of this DNA is 2 meter. Now this 2 meter, it will be very long to fit in a cell, to fit in a nucleus. Actually, this nucleus, all of these things, these chromosomes are present in the nucleus. So what happened? This nucleus is very small. It's just about 6 micrometer. Micrometer in sense how much? 10 to the power minus 6 into 10 to the power minus 6 meter. Now you may say that this is 2 meter. This is 2 meter, and this is very small. Nucleus is very small. So how can it fit there? How can it go and fit there? It's very difficult to go and fit inside the nucleus. That's why this all DNA, they are already understand that this chromosome is made up of two chromatin. So this is chromatin 1, this is chromatin 1, and this is chromatin 2. Now when they combine, they used to combine in a such, so for this is a combination. They get attached to each other. They combine in such a way by attaching each other. Now the point, see for example, let me show you here is a point. See, here is a point. Okay, here is a point. If we take a different color, if we take, okay, let me take this color. Now this point, this is a point where two chromatins join together, and when two chromatins combine, they are known as the chromosome. So in a chromosome, now this is a chromosome. Now for example, this is a, so now this is a what, chromosome. Now the place where they combine together, this is known as a, this is known as a centromere. So this is known as a centromere. So what is a centromere? If they ask you, so centromere is a point of a chromosome where two chromatins joined together. So this will be the definition of a centromere. Now this centromere, what is the function? They help in what? Help in attachment of chromatins. So this is also help in the attachment of the chromatins. Now another thing is that during cell division, when the cell want to divide, cell division means actually this is one cell. Now this cell will get divided and form from here. It will form two cell. One this cell and another is, maybe another is this cell. Now they form the another cell here. So cell division is what? Cell division is nothing but formation of cells from a single mother cell. We call this one as a mother cell. Now during the cell division, what happened is there is a, another term that we have to know is this, there is two chromatins. Now this is a chromatin, there is a chromosome, these chromosome are there. What will happen, the spindle fibers, these spindle fibers will get attached somewhere. The spindle fibers cannot attach in here. No, it cannot attach in there. It can only attach in the centromere. And what is a centromere? Centromere is a what? Centromere is a point of attachment of two chromatins in a chromosome. The point of attachment of two chromatins in a chromosome is known as a centromere. And what is the function of it? It is also serve as a place, as a point where the spindle fiber can attach together. Now, during cell division, what happen is this this spindle fiber will get shortened. This one will get short. It will become short. Once it becomes short, then what will happen, um, these two chromatins will get separated. In this way it will be separated, in this way another it will get separated. Why? This is because the spindle fiber is getting shortened. So as a result, uh, now this is a cell, for example. As a result, this one will move towards the this pole. Say for example, I am taking this one is north pole, for a north pole, and it is a south pole, for example. So one will move in this one pole, and this one is another pole. That means they are migrating in the opposite pole. And how does it happen? It is happening just because of the shortening of the spindle fibers. And where does this spindle fibers get attached? So it get attached on the centromere. And what is a centromere? Centromere is a point of attachment of two chromatins fiber of a chromosome. And what is a chromosome? Chromosome is a combination of two chromatin fiber, and they are highly coiled. Okay. Okay, another we will be talking about is in. Now suppose this is a chromosome. So chromosome always contain two chromatins. Now this is one chromatin and this is another chromatin. Now this is a chromosome. Now in each chromosome there will be some DNA nucleotides. So it will be so it is actually these chromatins are made up of DNA. This is also made up of DNA. This is also made up of DNA, and these also all are made up of DNA. Because in our first video we have already seen that chromatins are the composition of DNA and histone protein. So now suppose if we zoom at this one, and this is a DNA. In a DNA, now all the part of DNA cannot encode the protein. So all the part of DNA cannot encode the protein. Now suppose if this is a part of DNA which can encode protein, part of DNA, you can say DNA, a part of DNA also you can say, or a part of chromosome also you can say, because this DNA is a part of chromosome only. So the part of chromosome which can encode what? Protein. Which can encode protein of what type? Specific. It must be specific. What type of protein does it produce? It produces a specific protein. Then that part of chromosome is known as a gene. So we have understand already what is a gene. So gene is a part of chromosome which can encode a specific protein. Which talking about why do we need a new cell, or why do cell need to divide. So cell division means nothing but actually formation of new cells from a single mother cell. So let me give you, now suppose this is a one cell. Now from this cell they are forming two cells. For example, one this cell, one this cell I am taking, and another this cell I am taking. So formation of new cells from a single mother cell is known as a cell division. Now why do the cell need to divide? To understand that one, we have to talk about some new things. Number one is for growth. So if there are many things like this is maybe this is a small boy. Now this small boy need to go to a bigger one. He want to develop to a bigger one, and then more he want to become more bigger, for example. So all these things happen only because of the cell division. That's why cell division is necessary for growth. So number one is for growth. Now suppose again number two we'll be talking about. Now suppose there is a person accident. Now this is a person, for example, this is a person. Okay, he suddenly he happened to step on a big nail. Now this is an accident. Now what do you see when you zoom in here? You will see that in his feet there is some wound. There is some wound. Now to repair it, now what does it happen? Now if it is happening like this in his wound, in his leg, if there is an wound, will he be happy? It will be very painful. So actually to make it okay, what does you need? Again we need a cell division. We need to add a new cells, because the old cells is already damaged, so new cells is required. And for example, we'll be talking again in a human body there are many bloods are there. There are many blood. What are the blood? So blood is a stream of life, we know that. Without blood we cannot survive. Many bloods are also dying every day. Every day many bloods die, and new bloods are being added. What are added? New. So for this reason also we need to require cell division. Number one is for growth, second is for repair, third is what what is third. So after this person grow, he get married to a beautiful girl. For example, this is a girl. So this girl also will want to produce a child. So for another is for reproduction. So another is for reproduction. So why is cell division very important? If there is a question, then you can say that cell division is very much necessary for our life, because with that one it help in the growth. Another is it help in the repair of on our tissues. It help in the repair of on our tissues. It help in repair and replace. So this is under repair, and another is for reproduction also it is required. Thank you. Today I am going to talk about the cell cycle. First we are taking a cell, and this cell he want to go and divide, and divide means he want to form a two daughter cell. So this is a two daughter cell. So while forming one cell into a two daughter cell, now they just cannot form. Okay, I want to produce two daughters, so they just cannot happen. So it have to go a different type of process. So after that only it we will able to do. First, number one is, what does it do? So means number one, it have to grow in size. First it will have to grow. So this one is small, so this one, so after that it is becoming first this much big is becoming. Okay, this was small and later it is becoming a big one. So this is second part. So after growing, so this one we call as growing. After growing, what they will have to do? They will have to import, they will have to manufacture two things. Number one is they will have to produce RNA. They will have to produce RNA. So maybe this one I am taking as a RNA, big one. Maybe this one is RNA I am taking. Okay. So I'm taking this one is as a RNA, and another is also they have to produce a protein. So maybe this one I'm taking a protein. So this one I'm taking a protein. So please forgive me because of my bad writings. So maybe this one is I'm taking as a protein. Now this one is a protein. This green color is a protein. So maybe this one is under protein, this one is under protein, and this one is also under protein. So first, what is that? They are growing. After then they synthesis DNA, they synthesis RNA, and also they synthesis the protein. Another thing is that inside this one, when you zoom it, there is a DNA. So there is this DNA. This is single DNA. What they have to do? They have to make a Xerox copy of that. So Xerox copy, in science we does not say Xerox copy. We said that it is a replica. So they undergo the replication of DNA. So from one DNA it form a two DNA. So this is known as a replication of the DNA. After then only it will started forming two daughter cell, and these all things is taking place not in one step but in a several step, and then is predictable, and this is known as a cell cycle. So what is a cell cycle? It is the organized or the pattern that have to be going out, that have to take place before the new cell is forming. Now what is the difference between these cells? What is the difference between these two cells? Okay, so well, I have said that this is a mother cell, and this mother cell will be producing two daughter cell. Maybe this one, this is two daughter cell, this is one cell, and this one is two daughter cell. Okay, what it will form? Let's see. Actually, what is the difference between this mother cell and the daughter cell? To understand these things, we have to know the size. The mother cell have got a nucleus. This is nucleus, and the daughter cell also is having a nucleus. The nucleus is no problem at all. So it will be forming at these things. So what is happening? Just look at here. At these things, this whole these whole things, they are nothing but they are a cytoplasm. These all white things are a cytoplasm. So they are a cytoplasm. So they are the cytoplasm. So in this case, and this is a nucleus, and this is a nucleus, for example. Here we understand that the nucleus is more small, very small. The nucleus is very small as comparing to the cytoplasm. But in this case, if we look just the cytoplasm, the yellow one is a cytoplasm, the yellow one is a cytoplasm, and the nucleus is larger than the cytoplasm we have seen here. The nucleus is greater than the cytoplasm. So from here we understand that the main difference between mother cell and the daughter cells are the mother cells is having a big in size, and the daughter cell is having small in size. And also another one point, that is the one point, and another point is the mother cell is having a big cytoplasm, but in case of daughter cell, they are having a smaller cytoplasm. The nucleus is very small comparatively to the cytoplasm, and the nucleus here is larger as compared to the cytoplasm. So in this case, we can differentiate between the mother cell and the daughter cell. Now this daughter cell, what will happen? This daughter cell also will not stay just like this, will not stay just numb. It will want to become a mother cell. So at that time, what you have to do? It have to, number one is the size have to be bigger. The size have to be bigger. To become a mother cell, the size have to be bigger. And is so this one also have the size have to be bigger. So let me draw another bigger one. The size have to be bigger. Okay. Now, and the nucleus will also have to be a smaller one. The nucleus also will have to be smaller. So that's how the daughter cell become as a mother cell now. So this is a nucleus and this is a nucleus, and then let me draw again. Cytoplasm who said should to be a bigger one. Then only they can become as a mother cell now. So for that reason, this one and this one, this also they do. So in this way they become a mother cell. So what are the things that they have to undergo before entering to the cytoplasm? We would see. So number one is what they have to do. They have to import. They have to, first number one is grow. They have to grow. Number one is grow. Number one is they have to grow. Second two is they have to produce protein. Number third is they have to produce RNA. And another is they have to duplicate DNA. So it's very simple. So it's very simple that how to understand these things. Okay. So So because mother cell is bigger, so it is a bigger size. Now daughter smaller, smaller. Now how can they want to become a mother? If they grow and they have more protein and they have more RNA and they have more DNA. In that way only it is we can understand very easily. So thank you guys for attending my lecture. Now, next time we'll be talking cell cycle more in detail. Thank you. Student, now I'll be talking to you about the cell cycles more in detail. And first cell, phase, we see that it is a G1 phase. What is in G1 phase? In G1 phase, we need to remember that as soon as this G1 phase means a growth phase. It's a growth 1 phase, okay? Growth 1 phase. G for growth and it is for 1. So in this growth 1 phase, two things are happening. Number one is it will produce what? RNA. Another is protein. Protein will be produced. Now there is again this group 2 phase. This is G1 and this is G2 phase. So these two phase we have to understand what is the two difference. Now in group 1 phase, the cell have two options. Number one is either if they want to go for cell division, and another is if they don't want to go for the cell division. If the cell does not want to go for cell division, then it can stay for resting. It can undergo for resting. But in G2 phase, they does not have this option, because once it enter here, then it will go and surely have to produce a two daughter cells in the interphase. Now as soon as this G1 phase is over, then they will undergo in the synthesis phase. At this phase, what is produced is, at this phase, synthesis means who? As phase means they will produce only a DNA. They will produce DNA replication take place. As we understand that for the cell to produce another new cells, they have to undergo RNA, they have to produce RNA and they have to produce protein in previous video. So where is this RNA and protein produced? The RNA and protein is produced in the G1 phase and also in G2 phase. Once they started entering in the G2 phase, and after then the S phase, and here also of course there will be RNA and protein will be produced, but another new thing is that they will started producing a DNA. This make it different from the other. Now as soon as it enter on G2 phase, it will started still producing RNA and protein. More it will have to be produced, and once it is produced, now it will undergo in the interphase and then produces a new cells. Here they will produces a new cells. Cell division will take place. See, this is cell division that is taking place. So in this way it will take place, and this whole process is known as a cell cycle. And once it is reached the G2 phase, it will go to the interphase, and interphase then it will, the cell is want to go for the G1 phase, so it will have to grow on size, shape. All these things will take place in this process, and this, in short, this is known as a G2 phase. One very important question that can come here is, what is the difference between G1 and G2 phase? So in G1 phase they have got a resting period, and in G2 phase no resting period. And in which phase there is a synthesis of DNA, so you will write in a S phase, S for synthesis. And after G2 phase, what is coming? G2 phase, after G2 phase it will enter in the interphase. So this is the only things for the cell cycles, and it's very easy. So thank you and all the best. Cell division. How does the cell divide? So for cell division, we know that there are two type of cell division. One is mitosis and another is meiosis. And this mitosis happen, uh, mitosis is taking place in our somatic cell, and the meiosis is taking place in the sperm cell. So that is in the formation of the sperm. So this mitosis is are also of two type. Number one is karyokinesis and another is cytokinesis. First we'll be talking about the cytokinesis. So we'll be talking about the karyokinesis. And this karyokinesis involve in the division of the nucleus. So as we know that from the cell cycle, the cells which are ready for cell divisions will be entering in a interphase, that is the G2 phase. So this one is in G2 phase, and after G2 phase these things will be happening. So mitosis and meiosis will be taking place. So first and foremost, the first stage of nucleus division is the prophase. So what happened in the prophase? The the chromosomes are highly condensed, and what happened is two these two spindle fibers, steroids will be started moving towards the opposite pole. And once they have reached the opposite pole, uh, the microtubules, or you can say this is the microtubules, or you can say spindle fibers will be attached on the centromere of the this chromosome. So at this time two things is taking place. Most importantly, number one is the disappearance of the nucleus, and another is the attachment of the spindle fibers on the centromeres. Now if we go for the metaphase, then in metaphase what happened? In metaphase, all these chromosomes will be aligned in a equatorial position. See, they were aligned in this equatorial position, and then spindle fibers are still attached on the centromere, in the centromere. So second, after that what will happen is in the anaphase. Now in anaphase the chromosome will be getting splitted. This happened just because of the shortening of the spindle fiber. And then another thing is it will develop a furrow in this place. So furrow is something like this, as simple cleavage type. And then in telophase what will happen? Each chromosome will go and reach in the opposite pole, and once they go and reach the opposite pole, there will be a cleavage in the furrow. They will be clipped, and they will form a two daughter cell. And this separation of one cell into two daughter cell is involving to the division of cytoplasm. So that is known as the cytokinesis.